Just
It says it's preparing it.
okay. Something.
It says meeting is now streaming live on YouTube on my screen.
Thank you, Maureen. Okay.
Yes, it's good.
Okay. Okay, so welcome everyone for our seminar, our fine seminar today. So before we get started, we just want to acknowledge and thank you and give Jonathan Henshaw for his talk 2 weeks ago already about who cares and why, the coevolution of parental care and mating competition. And because I know some of you will run after the talk and would have chance to see, our next talk will be by Mikey Pardo, and he'll be talking about elephant vocal communication, a promising model for language evolution. And I also want to give you a little heads up on a survey that we'll be sending out this week, and we will appreciate your thoughts on the theme of the survey. Just keep an eye on it. Okay, so for today, we welcome Vincent Formica. He will be talking about population age structure changes, the shape of beetle society, and the landscape of social selection. So Vincent got his PhD at Indiana State University in 2001. And since 2012, he joined as a professor at Swarthmore College, where he is currently the chair of the biology department. In general, Vincent's lab is interested in understanding how the social environment can shape and how is it shaped by processes of natural selection. And he uses a variety of methods and approaches like selection analysis, social network analysis, animal behavior, and multi-level selection theory. And for answering these questions, he uses the forked fungus beetle as a model system. Well, if you go into his website, you'll see that he has a very, very impressive publication record and funding scheme, which I'm not going to talk too much about, but I would love to highlight Vincent's work. He's been very active and involved in evolutionary and international societies where he advocates for diversity, equity, and inclusion in science. And something that he's been really good at this, he's been the co-editor, of an interdisciplinary special section of the journal the American Society of Naturalists that is entitled Nature, Data, and Power, How Hegemonies Shape Biological Knowledge. So with no further ado, I would love to introduce Vincent. The mic is all yours. Thank you very much for accepting our invitation.
Hi. Hello, everyone. Thanks very much, Adriana, for that introduction. Let me just share my screen here. I'm getting a little bit of reverb. Oh, there we go. Okay, great. Okay, does that look good? Can everybody hear me okay? Yeah. Okay, great. Hi, everyone. Thank you very much for coming today. And like Adriana said, I'll be talking about population age structure and how it changes the shape of beetle society and the landscape of selection. Before I jump into my science, I'll advertise for just a minute that at Swarthmore College, this is in Pennsylvania in the US, we're hiring a full-time laboratory instructor. It's a pretty nice job. These laboratory instructors are faculty or instructional staff, and they help the faculty teach our courses, both in the lectures and the lab. And you get to work with a really great, gifted group of students. And so if you're interested, you should shoot me an email. I'd be happy to answer any questions, and that QR code will take you to the job ad. If you've got any promising master's students or even PhDs, we've got 2 PhDs in this role. It's a pretty nice job. Okay, great. It's nice to be in the US when you're hiring in science these days. It's nice to be hired into a private institution here. Okay, so I do also need to acknowledge my co-authors here. So my partner in crime, can you see my pointer if I do this? Adriana, can you see that? No? Okay, let me go back over here. How about that? You see that? Okay, great. So my partner in crime here on all of the forked fungus beetle stuff is Butch Brody at the University of Virginia. A lot of the work that I'll be talking about today is Phoebe Cook's dissertation work, and she actually started that with me as an undergraduate, and then went on to work in Butch's lab. So I got to work with her for close to a decade, which was really fun. Also, another graduate student who did a lot of this work, was really influential, was Robin Costello. And lastly, I'll give a shout out to Vitor dos Anjos, who was a undergraduate student of mine, who then went on to do his PhD in mosquito behavior at Princeton. But his ideas really largely shaped a lot of the work I'm going to talk about today. So this is not a group that I need to compel you to think that it's interesting, but the kind of goal of my research program is to kind of understand how complex social behaviors evolve. I think that one of the really fascinating things about social behaviors is that they happen in the social environment, right? It's made up of conspecifics. Those conspecifics have their own fitness, they have their own genes, and that creates for a really fun and exciting dynamic environment in which these traits are expressed, these behaviors are expressed, and under which they experience natural, sexual, and social selection. And so this idea of really trying to understand how things can evolve when the environment itself evolves has spawned really fascinating additions to Darwin's original theory of natural selection. So I'm thinking about things like indirect genetic effects, and the thing that I'm really interested in is looking at multi-level or group selection and how that might drive some of these complex behaviors like group hunting or termite mounds, or even something like duetting or parental care. So to capture some of the complexity of social behaviors, I've been using a tool called social network analysis. For those of you that are not familiar with social networks, let me give you a real quick look at these visualizations. So typically, these nodes or points represent individuals in the population, and a line between them represents some sort of measure of a social interaction between these two individuals. The thickness of this line can represent a lot of different things. For me, it's the number of times that individuals interact, but in some species, this could represent kind of the strength of that bond too. One of the great things about social network analysis is it lets us look at individual properties. So I can measure how well connected this particular individual is to his or her society. But I can also look at how I can compare the structure of these two networks to each other. So I know that this individual is really well connected in their society, but that society is not very connected itself, especially compared to something like this. We know that network position is really important in a range of species for a whole host of different things. So individuals that are well connected to a network are often exposed to greater disease, but they also tend to get information faster. We know that individuals in some parts of their societies have greater access to mating resources, and so on. We also know that the structure of the society can matter. So we learned a lot about this during COVID, right? Is that different? One of the studies I really like was about different universities and how fast COVID would spread through certain societies. And so we know that in animal societies, diseases and parasites and information pass much more quickly through societies that are structured with lots of connections. We also know that societies that are more densely connected, like this one, tend to be more resilient to disturbances, at least in their overall social structure. So one of the reasons that I'm excited about social networks is that somebody's interested in multi-level selection. I can measure an individual's properties about where it's located in the network, but also measure the properties of which it finds itself in, right, in the social environment. So this is just one way. I don't think there's anything magical about social network analysis, other than it's just a really neat tool to kind of boil down some of the complexity that we're seeing with social interactions. My lab's looking at this in a bunch of different ways. What I'm going to talk about today are really what factors in nature are contributing to the structure of animal societies. And I'll describe this work through my field observational studies, as well as some experimental work that we're doing. And then I'll touch a little bit on some data we have about how properties of the group feed back to affect the evolution of individual traits. This is something that we call multi-level selection. You can also call it social selection or group selection, depending on your background. We do all of this work in the forked fungus beetle. Their Latin name translates into the horned fungus beast. And we've been studying them for about 20 years at Mountain Lake Biological Station in southwestern Virginia. And we've captured, labeled, photographed, taken blood samples from, glued little numbers on, and followed about over 5,000 individuals over the past 5 or 6 years on a mountaintop in southwestern Virginia. They look kind of fierce in macro photography, but if you put them on your finger, you can see they're actually kind of charismatic and cute. Yeah. So these beetles, they live their entire lives on rotting logs and shelf fungi that are host to 3 different species of shelf fungi. Some cultures call these tree ears, and I just love that metaphor because they really do kind of look like ears. I call them brackets, like a shelf fungi. These brackets are the total resource for this species. So the adults eat it. The brackets serve as mating arenas where individuals will do courtship dances, combat with each other over access to mates. The females will then lay her eggs on the surface of this fungi, and then the larvae burrow into the shelves. They develop entirely inside the shelves. They pupate into adults. They're sexually mature, and then they chew their way out of the mushroom, and they emerge as adults ready to go. One of the nice things is that once they emerge, because they're beetles, so they're called holometabolist insects, they don't grow anymore once they age, which is really important for us to disentangle size from aging, which we'll talk about in just a little bit. Just a little bit more about their natural history. Forked fungus beetles are kind of a classic system to measure sexual selection and weaponry. So there's sexual dimorphism for horns. Males have these thoracic horns here, and these clapial horns in the front that they use for combat over access to the fungal resource, but also to females as mates. The females don't have horns. They just have these like little bumps on their head. They look kind of like a Princess Leia bumps on the top, and they don't participate in combat. We've tested that, and they don't seem to compete even for oviposition sites, which we found actually quite surprising. They have important social interactions. This is something I'm often trying to convince mammalogists of, that beetles can have important social interactions. This includes combat, as well as an extended courtship, and all of this happens on the surface of these logs and these brackets. The males can dance on the backs of females as part of the courtship ritual. We've measured it for 19 hours in a single pair, so it's quite an investment of the male to start courting a female. It lasts for several hours at the least. They tend to spend most of a breeding season on a single log. So we have very little migration during the breeding season, which is nice because we've got somebody who wants to study social interactions. I've got long-term repeated social interactions among the same individuals in one contained area. It's really nicely and easy to find. It's this log, and that's it. Like I said, they emerge from the fungus sexually mature, and they don't change size as they age. All of the work I'm going to describe takes place at Mountain Lake Biological Station. It's on top of a mountain, like I said, in southwestern Virginia. We have access to an old growth forest, which is really nice, some primeval forest. It's really important because the size and plentifulness of the dead trees really kind of limits the populations of these beetles, and we've got plenty of it in this old growth forest. It's currently owned by the University of Virginia, and it's the ancestral home of the Yasan people. I'd really recommend taking a visit. If you haven't been there already, you should stop by and see us and give a seminar. We love to have people visit. It's a really fun place to spend a summer. There's about somewhere between 50 and 100 people live there all summer long. So I'll take the time to kind of defend beetles as social creatures here and give you some of the examples of their social interactions. So I mentioned male-male combat. You can see from this photograph, this was one of my first days out in the field looking at these beetles, and I saw this social interaction, snapped a picture with my my digital camera, and was kind of hooked on this species. So this is a female here eating underneath a male, and so she's laying an egg while simultaneously this male is mate guarding her, and this other male is coming along and using his horns to kind of pry him off the back of that female and gain access and start courting that female. Like I mentioned, there's a pretty involved, at least for beetles, courtship dance where the male gets on the back of the female, faces head to abdomen, does his dance for several hours. Meanwhile, other males are trying to remove him. And they also just aggregate in kind of small to large aggregations around the edges of this fungi. They just seem to really love chewing on the edges. And so they're constantly jostling with each other and bumping each other around, and mating and courtship and feeding and egg laying are all happening within a few centimeters of each other on these logs. Hopefully this video will come through. This is a male combat that's taking place in one of our mesocosms. We call them beetlearies after aviaries. I'll show you, let's see some of this. Hopefully this will come through. It's a little shaky at first because it was being taken with an iPhone, but it'll zoom in just a second. And so you can see the males are using those clapial horns to try to pick up that other male to pry him off, and they're trying to push each other off of the fungal resource. In our mesocosms, they get a really good grip on that, and so they tend to fight a little bit longer than they might in the wild. I'll show you another picture. So this is looking up on the underside of a mushroom, underside of one of these brackets that we grow. And what you're seeing here is a really large female. Males prefer larger females. They have higher fecundity. And so there's a male on top. There's a really small horned male, and he's doing his dance on the back of the female. Along comes another male, and you'll see what happens in just a second here. So he's not going to use his horns, but he's going to use his bigger mass and his legs, as well as the hooks on the end of his feet to kind of push this smaller male off. And then after he does, he picks up that little male and throws him off into outer space. Now, in our beetlearies, that male would be fine, but in nature, that's where a lot of the mice and the small mammal predators are running around. And so there's a bit of a predation risk by being thrown off of the mating arena, but he's also removed from the population for several hours. And so he's missing at least that night's copulation attempts, on courtship attempts. One of the great things about this species is they've got a really easy to measure fitness component. So like I mentioned before, this courtship dance, this lasts several hours, and the female has complete control over copulation. So she's got a plate, part of her carapace called an anal sternite that protects her genitalia. So if she likes this guy's dance, then she'll open that sternite, and when they mate, the male will transfer a really large spermatophore that fills up the entire female's reproductive tract. And then he turns around and he mate guards her. At least we think this is mate guarding. So he now sits head to head, and he wraps his legs around the female, and he hooks some hooks that he's got into grooves under her underside, and then will guard her until she lays an egg, or at least for several hours. This has been shown to be a really, really good indicator of insemination success. So both in the field and the lab, if you grab pairs that are like this, 100% of the time, the females have spermatophores inside their bodies. And so we can at least say this is a pretty good sample of successful insemination. The females then lay a single egg on the surface of the fungi and glue it down with a frass-like substance. And then the single egg will hatch, the larvae will burrow into the fungi, and then live out the rest of its larval life inside the fungus. Let me show you a video of egg laying. This egg laying takes anywhere from a half hour to 2 hours. And so, just scan sampling is able to pick up all of these different behaviors. So you'll see in the back, there's a female here. She's doing that gluing process. So she's already put the egg on the surface, and she's using kind of like a hand-like extremity to kind of pat that down. And then this female in the front, you'll see her kind of pat that down here. This female in the front is also laying an egg and putting that glue on it. But while she's doing it, that male has climbed up on her back and is trying to court her and do a dance as well. So you can see there are also all these other eggs that have already been glued along the surface of the fungi. This is a pretty big egg. It's about the size of her head. And so it's obviously going to be quite a bit of a cost to not only lay the egg, glue it to the fungus, as well as the time commitment into gluing that egg down. Okay, so back to this idea of social networks. How do we collect these data in these beetles? So first of all, we go out and we run transects through our old growth forest. So it's about 2 to 3 square kilometers. And we run transects and we look for every population that has these species of fungi. We capture all of the beetles in the forest that we can. This is about 1,000, 1,500 beetles in a particular year. We bring them all back to the lab. We photograph them on a flatbed scanner. One of the great things is that they play dead when you breathe on them, when mammals breathe on them. So we just breathe on them and then you can put them on a flatbed scanner and photograph them. We take a little bit of blood, one microliter of blood, and that's enough to actually sequence their whole genome from. And then we cut out little size 4 letters. I hire students with very good eyes and keen fingers to cut out little labels. And we use dental epoxy to glue it onto their carapace, and that lasts for several years. Then we return them to their logs, and we hope that they think that the 48 hours they spent is kind of like an alien abduction, just hopefully a bad dream for them. And then we start scan sampling our populations 3 times a day for over 60 days. We record all social interactions and all behaviors. And this comes down to about it's anywhere between 30 and 40,000 observations per year because we're recording all social interactions or even solitary observations of these beetles. Something else I was going to say about that. No, I guess that's it. Okay. Oh, what I was going to say is that we use a proximity measure to indicate a social interaction. So if they're within 2 body lengths, or a body length and a half is about 5 centimeters, we consider that a social interaction. With social networks, there are lots of ways you can construct networks. So we at times have constructed just touch networks. So the individuals have to be in physical contact. You can construct just a sexual contact network. And most of the results are very similar, whether we use touching or 5 centimeters. But we think the 5 centimeters is pretty good because we're doing scan sampling, we're probably missing a lot of behaviors. And so the scan sampling we're hoping is a broader swath of interactions. Yeah, so this is what a lot of this looks like. Okay, so here's a social network of one of our populations, of our largest population this first year. So the blue individuals are males, the orange individuals are females. It's kind of hard to see what's going on in this kind of figure, but I still think they're useful to look at. One of my students described them as quantitative hairballs, and I kind of like that description. But you can see that there are some individuals that there's variation in connection. Some individuals are really well connected. They've got lots of thick lines, and other individuals are kind of out here on the periphery of the population with maybe 1 or 2 connections. One of our first observations, so we're sampling something close to 200 populations in a summer, but we have kind of 9 to 10 focal populations. Those are the ones we're going to 3 times a day and sampling over and over again. So here from our first year, here are just 9 populations that we sampled that year. You can see there's quite a variation in network structure. So even populations that are really small, you can see some of them are really well connected. And then in other populations that are kind of the same size, these 2 modules of the networks are connected by a single interaction, a single courtship event between a male and a female. Otherwise, we'd have 2 completely separate networks on the same log. Other populations really are these hairball populations where everything's just really, really well connected, even though it's a large population. So there's quite a bit of variation in structure. Turns out it's really hard to mathematically compare the structure of networks when they vary in size. And we'll come back to that in a little bit. Okay, but let's get some individual phenotypes here. I'm a guy who really my hammer is measuring selection, and so I like to use it on any question I come across. And so let's get some individual phenotypes here. And so one of the ways that we can do this is kind of mathematically distill an individual's location in a network and there's many of these what we call social network metrics. So this first one's called strength. It's a measure of centrality. It's basically calculated as a weighted measure of the number of partners, social partners you have, with some added partial interactions that every time you interact, you gain another strength measurement. So if you have high strength, you've got lots of interactions, lots of social partners, and lots of interactions with those partners. If you have low strength, you only have a few social partners and you don't interact with them very much. That's the one I'm going to focus on just because I think it's the simplest to wrap your head around. I'll talk a little bit more about another one called betweenness, which is a measure of how many different parts of a network you connect. How many different individuals have to pass through you in order to send a disease or a piece of information to another individual. So this individual has high betweenness because he's connecting these two parts of the network. This individual has low betweenness because he's kind of out here on the periphery of the population. There are other measures besides centrality. You can measure cliquishness, which is like a measure of how many of your friends are also friends, like how many triangles are you forming, but I won't talk very much about that unless you ask me some questions about it. So to be honest, I was very skeptical about social networks when they kind of came out. I thought maybe it was kind of going to be a fad in behavioral ecology. And the reason I got into it is a grant reviewer said, oh, it was for a totally different project, said, oh, you shouldn't be studying this. You should be studying social networks. And so I thought, well, and we got the grant was rejected. And so I thought, well, what I'm going to do is I'm going to go out, measure a couple of social networks and show there's no relationship with fitness. And that way I just never have to worry about social networks ever again. In order to measure selection on these network positions, I'm just going to use a standard Landian-Arnold type measure of natural selection, which is just a statistical relationship between a trait of individuals and the number of offspring they produce, or some sort of component of fitness. For males, I'm going to use insemination success. For females, I'm going to use the number of eggs that are laid. Right? And I'm just going to look for this relationship, and I'll do it in a multivariate space to kind of take into fact other traits that might be interesting. So I set out to do this to kind of show there'd be no relationship. You can imagine that that's not what happened. So here's some data. These are from our 9 populations. This is looking at horns. So we just wanted to confirm that we're seeing strong sexual selection in our population. This has been shown by people since the 80s, since Jeff Connor did the first work measuring natural selection in the wild on fork fungus beetles. And what we see is, I'll point you to 2 parts of these stats. One, there's strong relationship across all populations for thoracic horns. So individuals with bigger horns tend to have more insemination success. Individuals with smaller horns have lower insemination success. But you can see there's also no statistical interaction here among the populations. So the slopes of these lines are not significantly different from each other on the whole. And so what we're seeing is basically everywhere from moderate to really strong sexual selection for the size of those horns. So this is just confirming that we're seeing kind of consistent sexual selection in horns across our populations. You know, you're not seeing it in all of our populations, but overall, there's not a strong difference among populations. But when we look at selection for strength, we do see a strong, or we see a significant interaction between populations. So the relationship between strength and our subpopulation. So the relationship between strength and fitness differs significantly among our populations. And you can see that in some populations, we've got a strong positive relationship. So individuals that are really well connected to their populations are having higher insemination success. But in this population, being really well connected yields much lower insemination success, right? So not only are we seeing differences in the magnitude of selection, but also direction. And that's actually really rare. If you look at reviews of natural selection in the wild, often you see minor fluctuations in the magnitude, but rarely do you see whole reversals in direction of selection. So we do this for betweenness. We see an even stronger effect here is that some populations, this is a really strong negative selection and really strong positive selection as well. So again, what we're seeing is just differences in selection among these logs that are only a few meters from each other. And so there's something going on that is kind of driving these differences. And so we think it might be the structure of the network itself, or it could be something as simple as the density of the populations and the size of the populations that's driving it. So that's kind of the first result, is that we're seeing variation in network structure among wild populations, and we're seeing variation in natural selection. And so, of course, it was kind of disappointing, but also really exciting to me is that now I really did need to care about care about social networks. And so that's where it took my research from here on out. And so one of the things we wondered is like, well, what's leading to these differences in network structure, and what's leading to these differences in the selective environments on individuals' position inside of the networks? So I want you to just kind of hold that thought for a minute, because while I was chewing on this question and writing a grant and conducting some experiments to answer that question, along came an undergraduate in my lab named Vitor dos Anjos. And he asked a really nice natural history question as an independent project for a grant. He had gotten an internal grant. And he just wanted to know, do forked fungus beetles change their behavior as they age? So he looked at our data set and he said, oh, you guys have been collecting data for 4 years. And something I didn't tell you that Vitor noticed is that some of these individuals will live for upwards of 6 years. And so he said, you've got really nice data. You can look at cross-sectional analyses to look at, like, do young individuals behave differently than old individuals? But because you've got some individuals that are living for 4 to 6 years, we could do longitudinal analysis. And he really dove into the literature and came up with this question all on his own. And so we set out to answer that by mining our data. And so this is a really nice example of something a lot of field biologists do, which is just like collect all the data you possibly can, not knowing what you'll use it for, because along will come a collaborator that's going to ask something you didn't think of. So it's nice. In my papers, I get to say all data collectors were blind to these ideas of age because we hadn't even come up with the idea yet. So by definition, we were blind to these hypotheses. So what you might be familiar is when you start thinking about how age-related changes in performance, especially reproductive performance, right, is senescence. And that's what mammals and humans often experience, right? A decline in performance as individuals age. But in a lot of species, especially in birds and insects, we see actually reproductive improvement over time. So as individuals age, they get better. And so this was Vitor's very simple natural history question, is which of these do we see specifically in reproductive performance? And I won't go through all of his results, but I can tell you that the take home is that forked fungus beetles of both sexes improve reproductive performance as they age. So older males and older females court each other more, so they put in more effort in reproduction, and they mate more, so they copulate more. Males transfer more spermatophores, and the females become more fecund as they age. They lay more eggs. We've also done combat trials to show that older males are more aggressive and they tend to win more fights. And we've shown this by both cross-sectional data, but also tracking individuals through time, so longitudinal data. My dad, who's in his 80s, he really likes this paper. He reads all my papers, and he really likes this paper showing that there's just increased performance with age. He calls it the Scotch hypothesis, that you get better with age as you're a beetle. So Vitor was working on this while I was designing experiments to look at differences in selection and age structure. And together we kind of came up with this hypothesis, maybe the age structure of a population, you can think of this as the percentage of young in a population, perhaps that could influence the shape of social networks. So those of you that have taught ecology have probably used a figure like this, talking about human age structure in populations. We know that it affects things like the economy and GDP, but maybe this is a demographic factor. It's not very well studied in its influences on selection anyway, and definitely not on social networks. And so we started to ask that question, would it matter to the shape of the network, especially since older males are more aggressive, they're better fighters, and they tend to put more effort into reproduction. So we went out into our wild data and we looked to see what is their variance in age structure. Could that explain some of this variation in network structure? And so here's one example. You can see about 90% of the population are 1-year-olds, and maybe 10% are older than that. So 2, 3, and 4-year-olds. We do get some individuals that live up to 6, but it's not very common. And so in this population, it's pretty consistent. It's basically a really young population. But if I take another population from that year that's similar size, you can see that there's 50% of the population's young and 50% is old. The next year, it's as high as 75% young, and then the next year it returns to 50% young again, or 50% old. And so not only are we seeing differences in age structure within among our populations, but also seeing temporal changes across years. So just suggesting that there's variation in this demographic factor, and that might be contributing to some of these among population differences. But as I said, it's really hard to compare and look at causal effects on social network structure in the wild because populations vary in their shape, the species of host fungus, the sex ratio, the density. There's just too many factors that could be contributing and be really hard to isolate the effects of age structure. And so we did something a little crazy, is that we built artificial populations in the woods. They're artificial logs. And it took us a decade to figure out how to grow the fungi that these beetles live on. Nobody had cultured it before. Well, it turns out that one guy at the bottom of the mountain that we work on had cultured it, and he gave us a culture and started to teach us how to grow these mushrooms in captivity. So we call these beetlearies. It's a pun on the word aviary. These are screened enclosures with artificial logs where we control the size and shape and genotype of the fungi. We control who goes into these populations. So all the populations have the same sex ratio, the same densities. We control the distribution of body sizes and horns. And we breed the fungi the beetles and the fungi that go into it. So we know the histories and the ages of all of these individuals. Yeah, so here's what it looks like. We do the same methods we did in the wild. We label these beetles, and then we put them into the population. We do scan sampling and record all of their social interactions. Like I said, it took us a decade to figure out how to grow these fungi, and now we're doing it on an industrial scale. So this spring, we're starting a big experiment. We'll be growing 1,000 mushrooms. And so it's a full-time job of one of our lab technicians. And we learned some of these practices from actually from Chinese traditional medicine. They grow this on an industrial scale. This is similar to a mushroom called reishi, which is very popular. Some people call this particular species we work on Appalachian reishi. And you can buy it in grocery stores for dollars per pound. And so we always wonder if maybe we can get a side gig to fund our research by growing these fungi. Okay, so here's our experimental design. We've got 12 of these enclosures. So that allows us to have 6 populations of 2 different treatments, whatever treatments we want. And so we're going to put 36 individuals into each beetleary. We've got 12 of these, so it's 430 beetles. And so for this particular experiment, we came up with 2 different treatments, a young population and an old population. And so the young population here is made up of 1-year-olds and 2-year-olds. The old population is 2-year-olds and 3 and a few 4-year-olds. So young skewed, old skewed. And then we surveyed them 63 times over 3 weeks. We're measuring, again, their social environments and their fitness. So I'll give you a look at this age structure. I don't know if you've looked at enough social networks to kind of see this. These are the young populations out of our beetlearies, and these are the old. Not immediately obvious. There might be differences in structure. Let me show you 2 examples kind of the extremes. You can kind of see what's going on here. The younger populations tend to be more connected. So it might look more spread out, but that's just the visualization. What you're seeing is lots of connections, lots of heavy social connections, lots of interactions between individuals. The old populations are more sparsely connected. And so it turns out that no matter how you measure these populations, you get that same answer. We can look at tie density, coefficient of variation, clustering, path length. These are all measures of the whole network, and they're different no matter how we measure these populations. Younger populations are more connected. What's very cool, I thought, is that the total number of social interactions is not significantly different between the young and old. So it's not just that the old are not moving very much and they're just not interacting. It's that they're having the same number of social interactions, but the structure of the population is really what's differing between these two treatments. So we can also ask, within populations, is there structure? And the answer is yes. So let's zoom in on just one of these old populations now. You can see that all of these young individuals that are colored the lighter color are all interacting with each other, right? And that turns out to be statistically too. Younger beetles tend to be more central to these populations and are more connected, both in the young and the old populations. And so another way to say this is that individual age is positively correlated with partner age. Old individuals are interacting with a few old individuals. Young individuals are interacting with lots and lots of young individuals. Then Phoebe, kind of in a post hoc analysis, was really curious if this change in behavior from young to old is something that happens over a beetle's lifetime, or if there was something like the hidden fraction going on, that maybe all of the really highly connected beetles are dying out. And so she realized that a lot of the beetles we had used in this experiment in 2020, we'd actually used in a very similar experiment with identical conditions in 2018, testing something else. And so what she did is she looked at just the number of social partners that an individual has and showed that compared to a random sample of individuals of 1-year-olds, and then she did it again in 2020, a random sampling of 1-year-olds, we're seeing a change in individuals. So as they age, they are decreasing the number of social partners that they have. They're becoming less gregarious, suggesting that this is really something that changes with an individual as they change. So the last question we asked with these data was, how does population age structure affect the selective landscape? And so we're going to do this in what's called contextual analysis. This is kind of a classical multi-level selection analysis. And I've been really careful to try to not to overconsume these data. And so we thought really carefully about how to run these data, and we've run them once. One of the problems is that you've got to do permutation. So these are generalized linear mixed models where the p-values are calculated through permuting, doing node-based permutations in the network. I'm happy to talk more about that. But we ran 2 separate models, one for males where we're going to use for fitness, we're going to use number of insemination, and one for females where we're going to look at the number of eggs that were laid by that particular female that were observed laying. We think that this is a pretty good measure of female fitness. We think we're seeing somewhere between 10 and 20% of the eggs that a particular female is laying. We know that because we can count up all of the eggs on the fungus brackets, and we can look at how many times we watched females lay eggs, and we can say we're seeing a small subsample of the eggs that are being laid. Okay, so we're looking at things like body size, but then we're going to look at age across 3 different scales of organization. So an individual's age, are they young or old? Then we looked at the average age of their social partners. And we know from other experiments that it's really male, or it's most likely male interactions that are driving fitness effects, both in males through male-male competition, but also in females. So we know that from experiments where we've looked at fitness effects of males on females. So because our data, we've got 400 individuals, but this is a pretty packed model, we decided to just look at the male social partner age. And so the effects of the males that you're interacting with. And we know there's some covariance here, right? We know that young individuals are largely interacting with younger individuals. And now we've got our population age structure, and this is a treatment that we've applied to these 2 sets of populations. So individual, your immediate social group, and the age structure, young or old, of your population. We're looking at selection on individual strength. That's those data I showed you before from the wild. But then we're interested in, does our treatment explain a lot of that variation that we saw in selection on network connectedness or strength? And then we've got a series of random effects to control for population and other grouping variables that we have. Okay, so here are the male results. So first of all, it's really nice. We were able to recapitulate our results from the wild, is that individuals, this is insemination success, they tend to do better as they age. Interestingly, these are much higher resolution data because we can find everybody in the populations every time we go in. And so what we're actually seeing is not only reproductive improvement, but actually a decline as individuals get older. So this is commonly referred to as a middle-aged effect, in that maybe it's these middle-aged, these 2-year-olds that are having the highest fitness. So I like to tease my dad about this, that maybe beetles, actually, it's the middle-aged beetles that are doing the better. But it's nice we were able to recapitulate that kind of changes that we saw in the wild. Okay, so this is what we call social selection. So these are traits of your immediate social partners that are affecting focal individuals' fitness. And so what we find is that individuals whose partners are older tend to have lower fitness. And this is what we expected, right? We know that older partners tend to be more aggressive. They tend to put in more effort into mating. And so if you're a male who interacts with lots of old other males, we think you're going to have lower fitness, and the data bears that out. There was no significant interaction here. So both of these treatments, we saw it in both the young populations and the old populations. You interact with older beetles, you don't do as well. We saw no differences in the average mating success between our two treatments, which is good for us. There's not group selection here, so I don't have to try to explain that to reviewers that don't believe in group selection, which happens from time to time still. And then lastly, we were really surprised in that our demographic treatment of age structure doesn't seem to explain any variation in selection for network connections. So individuals we saw in both populations, both treatments in all of our populations, as individuals become more and more connected to their network, they tend to have higher insemination success. I should mention that all of these measures of the networks do not involve mating interactions. I've removed them from the data set. So I'm really just looking at your place in the society. I didn't want to have any autocorrelations here. So this is not kind of dishearteningly explaining that variation we saw in selection and network strength. It must be something else that's describing that variation. Okay, so that's males. So I was really excited about these results, thought it was pretty cool. But then we looked at the females, which is not something we published in the wild, although we did do the analyses, and it was similar to what it was in males. We see again that females, so now the reproductive success has switched to the numbers of eggs laid. And we see what we saw in the wild is that first-year individuals don't do as well as older individuals. And we start to see this decline in females as well, but it's not significant. In females, we see, again, the same trend, but it's not a significant result. So the older sets of males that females interact with, they don't seem to lay any more eggs. So no social selection in females. But interestingly, what we did see is that females in older populations lay fewer eggs than would be expected once you control for individual age. So this was a bit of a head scratcher for us, and it might be for you too. You might be saying, how is that possible? These old populations are made up out of 2-year-olds and 3-year-olds. So how are they laying fewer eggs? Like, these data don't look like they match. And that's where I have to remind you, like I had to remind myself, that this is in the same model. So this is the effect of your treatment being in a young population or an old population after you've accounted for variation in the eggs being laid based on their age, right? So this is really the effect of the treatment after controlling for the fecundity benefit you get as you get older. So, whether we're going to call this group selection or not, I think is something we're still scratching our heads on. It's not a really strong effect. And so I'm not jumping up and down and saying I'm seeing really strong group selection in females. But it is really suggestive that that age structure may have some effects on the groups, especially in output of number of individuals. But here's the real fun answer that we saw is that we saw the same trend we saw in males in the old populations. So in old populations, if you're a female and you're really well connected, you lay more eggs. But in the young populations, it doesn't matter. There's no selection for network metrics when you're in a young population. So this is what I call changes in the landscape of selection. So the demography is affecting the strength of selection. So what do we think is going on here, right? This was kind of, like I said, a head-scratching moment. So we think that age structure alone may not be describing selection differences we saw in wild populations. What's going on with the females? We're not quite sure. Even though this is an experiment, we don't have the resolution of the data to really know the mechanisms of the behaviors that are happening. We think that maybe I should point out that these are relativized strengths. And we know that older populations are less connected. So maybe if you're in a sparsely connected network, being the top connected individual might be really good. But when you're in the young populations, everybody's connected. Being the top connected individual just doesn't gain you anything because there's some sort of threshold over which it just doesn't matter how many connections you have. It could be that maybe in younger populations, there's some sort of trade-off between being really well connected with male courtship and harassment. And so we know that females that are in really well-connected networks tend to get courted a lot more. And we know that males prefer older and larger females, and they tend to court those females a lot more. So taken together, I mean, something we're wondering is, could this lead to sexual conflict in the evolution of gregariousness, or help maintain diversity in gregariousness if your demography shapes selection on females for gregariousness, but not in males? Or is this difference we're seeing between the males and females really driven by the differences in the fitness components that we're measuring? We're unsure about that. So something that we're really interested in and starting to develop a kind of holistic model to look at is thinking about the ecology and life history factors that might change the age structure of social groups. Something like predation could make everybody young if ever at the end of every year everybody gets eaten, and that might cascade to both individual behaviors, the individuals' network positions. It could shape the landscape of selection. It could be driving group network structure, which I showed you it does. The age structure drives group network structure, and possibly driving group selection. So should we be adding, this is where we're still kind of wondering, should we be adding the age structure of a population to our pantheon, if you will, of demographic factors like OSR and density? Or is this just kind of another demographic factor that's just another source of environmental noise? I would say the jury's still out on that and will require a lot more experiments that we have planned for the next few years. I always tell my students that you should be able to sum up your talk in something that used to fit in a tweet. I'm choosing the less fascist option now on Blue Sky. So here's my Blue Sky tweet here. Age structure may be an important and understudied demographic factor that shapes animal societies and behavioral evolution. I need to thank my social network, and because I teach at an undergraduate-only institution, it is definitely skewed towards the younger side of things. But these are really important collaborators to me, as I showed you with Vitor, kind of generate some of the ideas that I take and use for the rest of my research program. And with that, I'll take any questions. We're going to have a discussion.
Thank you very much, Vincent, for such a great talk. It was really, really fascinating to hear about this beetles. And so I open the floor for discussion. So remember to just type in a question mark if you have any question. If you're a student or a postdoc, just put an S next to your question mark and I will call you first. And if you are on YouTube, just make sure to type in your question and we'll make sure to have it here. Okay. Well, so far, we don't have an, Vince, can you unshare your screen so we can see each other? Yes.
That's great.
Michael, you have a question.
Thanks very much. This looks like a really intriguing system and a very nice study combining field data with laboratory experiments, or say semi-natural environments. I enjoyed that very much. I have a question regarding your information about the differences in population age structure in the field, or the reasons for that. I mean, is that simply mortality reasons, or does it have anything to do with the dispersal differences between different populations? Do you have any clue what really causes this apparently quite strong difference between populations that seem to be very close by? So I would expect that the ecology is quite similar. It's quite surprising, isn't it?
Yeah, it is really quite surprising. We were very surprised by the variation in age structure, and it's not just those two populations. It's quite pronounced across the whole meta population. I have some ideas. I don't have good data to back any of it up. So that one population I showed you where it was like 90% young, there's actually a family of mice that live in that log. And we find they eat the beetles like a oyster on a half shell. They pop the elytra off and they leave the tag on the elytra on the floor, and we can find it with a UV light. There's also a chipmunk that lives near that population, and we have video evidence of him popping the elytra off, and he leaves them in a big big pile for us to find. So I know that there's a pretty high predation on some of these individuals because I find their dead bodies. I think that plays some role in it, and I'm interested with our long-term data at looking at how mast years might affect increases in small mammal abundances, and that might shape some of this temporal variation. If everybody gets eaten in one year and the next year the mouse populations crash, that's there. So it might be predation. I also think so. The fungi that are the host to this population, they are also senescing. And so within about 5 years, that individual, depending entirely on the size of the log, is senescing. And so individuals, the beetles can take anywhere from 1 year to 4 years to chew their way out of the brackets as larvae. They can just spend a lot of time as a larva. And so there might be, I think there's probably interesting extinction and colonization effects about how old that individual is, the size of the log that it's on, and the kind of history of that population and how long it's been there. And so if it's a new population, we know from earlier work that young beetles tend to disperse from their natal environment after they eclose. They're more likely to leave. And so if there's a new population that the fungi just sprouted, I suspect that would be younger. So I think there's a whole host of ecological factors that are kind of contributing to this demography.
So apparently you also looked into dispersal because you have the data of these marked individuals. Do you see any change with dispersal propensity with age?
That's a really good question. We have the data, but we haven't been able to run the models yet. We've started to do that, but we've only collected really good dispersal data that I'm happy with over the past 3 years. So I haven't had a student yet who's really wanted to dig into that. The dispersal data I have was actually from a dissertation by a student of Henry Wilbur's at the biology station before I even started. And she did a really nice mark recapture study, but the data's in the gray literature. It's kind of stuck in the dissertation. But what she showed is that there seems to be younger beetles tend to disperse more, but she didn't track individuals late into their life. So we don't know if older individuals disperse more often. They do keep their wings and they're able to fly when they get older. I know this because if you put them in solitary confinement and then try to do combat trials, they fly out of the little arenas we put them in. So they can fly late into their life. There are some beetles, right, that kind of digest their wing muscles away, and that doesn't seem to be the case.
Last brief question, sex differences in dispersal?
Sex differences in dispersal. Not that I remember from that dissertation, but stay tuned. Once I get a student who's interested in digging into that, I'd be really interested to see what's going on. That would be really interesting. What I remember from her dissertation is there was a small and significant difference in dispersal, but not one that we are sure was biologically relevant.
Thanks very much.
Yeah, thank you.
Thank you, Michael. Yeah, just a reminder for everyone, just state your name and your affiliation and perhaps a little bit of what you work on, just, to introduce to the folks that are new to the community. So we strengthen our community. I don't know, Michael, if you want to introduce yourself again, just briefly.
Yeah, sorry that I forgot that. Michael Taborski, University of Bern. I'm working with social behavior, the evolution of social behavior and cooperation with a number of different model systems, including beetles also, ambrosia beetles in this case, fish and rats.
Oh, thank you. Okay, so next question comes from Carolina Morales.
Oh my gosh. Now, hi everyone. Thank you, Dr. Formica, for the talk. It was very nice. My name is Carolina. I am finishing my PhD in ecology at the university from Pontificia Universidad de Chile, basically, and I work with social behavior, basically working with Luis Evensberger on social instability and effects of cooperation in Octodontigus, a rodent native from Chile. I had a question for your methods because you said that you capture every year like something like 1,400 beetles, but as they live multiple years, you have an increasing database every year, right? Or you lose a lot of them, or how does that work? Because I don't imagine like all of them being like dispersing or dying, or yeah.
Yeah, great question. So this is something I skipped over that's actually pretty important. And so we have about, this is a rough estimate. I need to really run these mark recapture models, but we have about a 50% mortality rate among individuals in a year, in any given year. So that 1,400 number includes the individuals that survived from the previous year. That I was saying. The way we age beetles is kind of a brute force method. So we've got these focal populations that we're studying social networks in. And the way we get it is the way we get age is that we sample everybody within 100 meters of those populations, which is twice the dispersal distance. And so we just hit all of the populations in the entire forest. Sorry, let me turn off my phone. And we hit every single population in the entire forest at least 3 to 4 times over the summer. And that's enough to get something like 80 to 90% of the beetles in a particular population. And we just mark all of them. And then anybody who shows up unlabeled, we're assuming is a newly closed individual. So that's going to lead to some error where we're going to be we're going to be estimating that some individuals are young when they're older, but that should lead, we actually think that should lead to less age effects than what we're seeing. And so if we're making that error, we're hoping it's not getting in our way too much to detect age differences. But yeah, there's no nice way to get like tree rings or look at the size or something and determine the age. We just have to do it through brute force. That's another reason we moved to the beetle areas where we know the age because we know how old that beetle is because we found it in our cage.
And how many years you have all this data for?
We have data in the field for 5 years, full data sets for 5 years. COVID really kind of broke our system. So we have cohort marked population, so no social data for an additional 3 years. And then just last year, we gave up on the field data because we didn't have grant money and the other things had happened. So now we've moved entirely to beetle area data and these experiments because we think we're able to kind of get at the questions a little bit better. But that field population is always calling to me. There's still individuals we marked 4 or 5 years ago living out there with little dots of paint on their butt, and I'd love to go back out and catch them. Yeah.
Okay. Thank you so much.
Yeah, thank you.
Thank you, Carolina. I'm going to call Linda Lani and then David. Okay. So Linda Lani.
Okay, so hi everyone, my name is Ndelani Maguya, and I'm currently a postdoc at the Succulent Karoo Research Station. And my general focus is just trying to understand the costs and benefits of solitary living using the bush karoo rat as a model species. So my question was, I'm sorry if I missed it, but what was the relationship between age and size of the beetle? One would expect that larger individuals are older, but was this really significant? Like in terms of body mass, was this really significant in your models, or did you control for it or not include it at all?
Great question. Yeah, so the beetles, fortunately for us, the beetles, once they eclose, so once they pupate and go from pupa to adult, they eclose with their final carapace. And so they stay the same size no matter how old they get. So an individual that's 5 years old is the same size as he or she was when they first eclosed from pupa. So we do put that body mass into our models, but it's more about looking at weaponry and for females, the males, there's strong male choice for larger females. But that's why we're putting that into our models, not worrying about age, which is nice for us, right? Because then I don't have to try to disentangle them as co-varying. Body size we think is probably due to something having to do with the nutrients, both genetics obviously, and some of their environment, and probably really heavily on the environment that they're getting in the fungal resource where their mom laid the egg. And that's been true in a lot of other beetle species, and that's been true from our lab work as well. That if you give them really crappy food or not enough of it, they eclose really early and they're micro beetles. So yeah, so it's probably more environmental based and it's not age based.
Yeah.
And we have had some beetles that get injured, so they break their horns and then they live for 4 more years with that broken horn, and is really kind of interesting to track that. That's actually what got Vitor really interested. He found these beetles with broken horns, and he was asking me like, "Oh, do you know this beetle?" I was like, "Oh yeah, this beetle MOV, I've been tracking for 4 years now. I wonder how he's going to do with this broken horn." And he was like, "Wait a minute, you know these beetles longer than you know some of your students?" And I was like, "Yep."
Yeah. So just to follow up, did you do, because some of the females are quite large and they were like laying bigger eggs. Do those eggs then become bigger individuals, or is there a correlation between the size of the egg and how big the individual gets after it pupates?
Oh, good question. I don't know the answer to that. We've done some stuff. The problem is that they're kind of a pain to grow in the lab. They take a lot of care. And they take a year and a half to reach maturity at the fastest. And if I do that, they tend to be kind of small. And so I'm just not trusting those data. We've started growing them in mesocosms in cages in the wild, but we're doing them in groups to make it easier to do the husbandry, because we need thousands of beetles to get those 400. And so I don't know the answers to that. Again, I'm waiting for a student to come and ask that question: if larger females lay larger eggs, and does that result in larger offspring? I don't know any of that stuff. We know very little about the larval biology of these beetles, actually. We know they are cannibalistic, but we have no idea how that contributes to size either. So all of that is a mystery and is important, because size is driving a lot of these things, and we just don't know much about it. Thanks.
Thank you.
Thank you, Vindalani. David?
Hi, Dave Queller from Washington University in St. Louis. I work on social amoebas, both their social interactions and their interactions with bacteria, positive and negative. Nice talk and really interesting system. Early on in the talk, you mentioned your initial skepticism about the network approach, and that kind of resonated with me a bit. But then you said the source of your skepticism was that you didn't think there would be selection on the network characteristics. And I'm not especially surprised by that, which is not to take away from the interest of actually finding it. But what bothers me a little bit is what it means biologically when you find these things. I understand well what it means when you find selection on male body size or horn size or something like that. It's a little bit less clear to me what it means when you find selection on network strength or network connectedness, something like that. And so that has two parts, I think. And I think I'll ask the first part and let you respond to it, and then come back to the second one. And the first part is just that, repeating what I just said, I understand normal traits, selection on those easily, but when you move to network traits, you both moved into a social arena, which doesn't disturb me because I work on social selection too, but also rather complex social traits, interactions with lots of individuals at the same time in different ways. And so what does that mean in terms of the way we usually think about adaptation? What does it mean for adaptation?
Yeah. Well, I'm not surprised, David. That's the $1 million question, right, about social networks. And this is what I really struggle with about, like, okay, how do you have an evolutionary response to selection on network traits? What is it? Is it real or not? And so I'm assuming that a network trait like your betweenness or your strength is an aggregate measure of a whole bunch of individual traits that are either just a composite measurement. I kind of think of it, this is not a good metaphor, but I kind of think of it like a principal component measure, but I'm actually measuring it instead of like creating it from linear combinations of a trait. So maybe it's gregariousness and aggression and some measure of a social experience, right? And so if you're selecting on betweenness, maybe you're actually selecting for movement and home range and gregariousness. So maybe that's what's going on. There are folks that argue that these are truly emergent properties that cannot be reduced to smaller traits. That's not really how I think about it. I think about it as kind of capturing aggregate behaviors that we might otherwise be missing because we're not thinking of that behavior and what it is and necessarily measuring it. So that's how I think about it. I think if you're going to get an evolutionary response, it's going to be because it's the product of smaller traits that kind of cascade up to this thing that I measure that we've happened to call strength.
Okay. I mean, that makes sense, although it would make more sense to me if you were able to back up to the smaller traits in the end. But my second part of the question is, if you're thinking about selection on these things, it's a social trait. You can think of it in terms of group selection. There is a group selection coefficient, if you like. But what about the heritability? So if you're interacting with non-relatives, the simplest way of looking at it is that those interactions are not going to be heritable. But they might be if there's positive or negative assortment or something like that. But either way, the evolutionary response doesn't depend just on the selection coefficients, but also on the heritability.
Yep. Again, great question. So there have been some folks that have been able to use animal models to show there's heritability for network position. And then there are some folks that have done GWAS showing that there's some genetic basis for these, although those tend not to be in the wild or they're done on humans, and I get a little squishy, a little worried about GWAS in humans for social network traits. But yeah, that's a really good question. We know that it's a repeatable phenotype, so I can pick up an individual and move it to another population with different individuals, and I get a very similar traits. Is about repeatability in these things is somewhere around 0.2, 0.1, 0.2. So something similar to kind of foraging behavior. But that doesn't mean
heritable.
Right. Doesn't mean it's heritable. It's the first step.
It probably is if it's repeatable in different contexts.
Right. So two things I'll tell you is that it's repeatable in different contexts, and I can take individuals. An experiment we did is we let the networks run in these beetle areas. We took individuals from individuals that were kind of on the outskirts of the society, and we put them all together in populations. And then we took all the really gregarious ones and we put them together, and that was still repeatable in those different contexts. And the networks were the emergent property you would expect. The non-gregarious populations, they were all loosely connected, and the ones we put together, they were all really tightly connected. So there seems to be not only some repeatability, but some robustness to being even, we thought there might be kind of compensation for being in an all shy group, but we didn't see it. So yeah, so it's suggesting there's some heritability there, but I'm certainly not claiming it yet. We've just sequenced the genome, and we're writing a grant to sequence all the genomes of all 5,000 individuals, and then we'll be able to do some heritabilities and maybe some GWAS to see what comes up, but. That's the next step. We collected blood from all these guys, and we were really surprised we can use human forensic kits to sequence the whole genome from 1 microliter of blood, which was just like amazing to us.
Thanks.
Yeah, thank you so much. Those are great questions.
Thank you, David. I will call Lily and then Suleiman, okay? Just to give priority to the students.
Hi, I'm Lily. I'm a PhD student at Harvard studying how motherhood shapes patterns of female sociality in bonobos. And I was actually one of Vince's students, like so long ago. So I actually was there right before you started the beetle areas. So actually my question is kind of tied to something that you might have learned from the beetle areas, which is the context of these interactions. So something that I think of when you mentioned the strength of these interactions and the networks is like socio-ecological factors that might, drive females to associate and then drive males to associate around those females for mating opportunities, and how you might be measuring social interactions that are actually just a result of both like resource interests for females and mating interests for males. And it's just that if you get around a female, you're going to interact with more individuals, but you're also going to have a greater likelihood of accessing those mates and therefore having greater reproductive success. So do you have an indication of how these interactions are patterned to speak to whether these interactions are happening outside of just a mating context?
Oh, that's a great question. So this reminds me of kind of Schuster and Wade's mating strategies book, right? Is like the resources are out in nature, the females go to those resources, and then the strength of sexual selection is dependent on the kind of placement of that resource and whether males can defend the resource that females are attracted to. Yeah. This is something that actually in Schuster and Wade's book, they actually say fork fungus beetles randomly. They say fork fungus beetles might be a great system to test this in. And so ever since I've read that line, I highlighted it as an undergrad, and I've always wanted to go back to it. So the short answer is we've got some clues, but we've not done a systematic study. So we have an experiment. The first experiment we did in the beetle areas is we changed the pattern of the fungi. So we put them, that was that experiment in 2018, we put them in clumped areas and another one where we dispersed them. And we were wondering if that contributed to differences in network structure. And it does, but not a lot. Matter of fact, I think the editor suggested we title the paper like surprisingly little difference in effect of ecological placement. So I don't know. So the answer is it does change the structure. It changes group selection on females. It does not change individual selection on males. So again, we're seeing this kind of group selection or effects of the network structure on female success. Yeah, so the other area where we're looking at it is looking at home ranges of males and females and visits between males moving to those brackets, and whether there's different strategies for males with long and small horns and how they move from bracket to bracket. But I've not done that analysis. I'm still trying to wrap my head around how I quantify that type of resource defense, because the males just move all over the place. They have home ranges that vary in size. Some are small, some are the whole log, but they don't like defend territories at all. But they do fight over access to it looks like over access to these populations. So the answer is I don't know, Lily. It's a good question. Yeah, and I just need to dig into it a little bit more. Yeah, but I will give you a shout out for collecting some of the data that went into our analysis there. So thanks.
Yeah. Thanks, Vince.
Yeah, thank you.
Thanks, Lily. Suleima?
Yes. Thanks. Suleima Tang Martinez. I'm retired from the University of Missouri in St. Louis. And before my retirement, I worked primarily on social behavior and chemical communication and various aspects of that, primarily in rodents. And I also, later on in my career, started working on conceptual issues and particularly around sexual selection. I also have an interest in the history of animal behavior and biology more broadly. I have several questions, and one of the questions I had you just answered, which was great, which was about movement and range size, and you answered everything I had. I'm a little confused because I realized that as you were speaking, you mentioned the very lengthy courtships with the males dancing on top of the females, and then the males mate guarding. And then you said the females, after that, deposited one egg. And initially, I assumed that each courtship event led to only one egg being deposited by the female. But then as you went on, I sort of had the feeling that that wasn't correct. So maybe if you could clarify that, and how many males do females mate with? Does that make a difference in terms of their reproductive success? Those sorts of things. But I also want to say, I can't remember if I already did. I just thought the talk was totally fascinating. So yeah, can you understand? Are you understanding what my question is?
Yeah, totally.
Okay, great.
Yes. Yeah. So yeah, I see what I said and why you might think it was one egg. No, We thought that originally in the original papers, that's what they suggested, is that each mate guarding led to one egg. That's certainly not the case because females store sperm. So we dissected a whole bunch of females, and they have these really cool reproductive anatomy that stores the sperm, and the in beetles, and especially in ours, the sperm embed themselves, the heads, into the cells of the female's reproductive tract, and they can keep them alive. And we had one female that we kept in captivity for a year and a half in total isolation, and then we gave her some fungus, thinking, okay, this female hasn't mated, she hasn't laid eggs, we're okay. We gave her fresh fungus, she started laying eggs. So clearly they can store sperm for years, or at least a year. And so no, I don't think that there's one egg per mating. That would be super nice and easy to talk about, but no, I don't think so. And so, that is another piece about whether even male fitness as insemination success even measures their actual reproductive success. And now that we've got the genome sequence, we can start doing some genetic parentage, and see if it matters. Your other question was kind of like nodding a little bit towards like a Bateman gradient. So do females who mate with more unique males tend to have more offspring? We've not seen that in this species.
Okay.
But we haven't done molecular parentage to answer that question. And as you may know, molecular Bateman gradients have been called a little bit into question because there's some idea that you may just kind of always detect them based on sampling. So we haven't even asked that question yet, but it's a good one. Yep.
Yeah. But females do mate with multiple males?
Oh yeah, multiple males, some on the orders of tens of multiple males, upwards of 20. Some only mate with one. Males are the same. So, anywhere from 0 to 1 to 20 different females. Yep. And then we've had, because when you stare at things long enough, you can see just about anything. We've seen one pair that only mated with each other in a crowded population, like 20, 30 times. So.
Yeah. But you're saying that in terms of the Bateman gradient, you're not seeing any effect on female reproductive success as a function of the number of different mates that she has, correct?
That's correct. The last time I did the analysis was on 1 year. And so something I'd like to do is take the full data set and ask if we see Bateman gradients for both males and females. I'm waiting to do that until I have the molecular data, because I just don't think it's a good idea for males, because it would just be an autocorrelation. Yeah.
Okay, thanks a lot.
Oh, thanks for your question. There's a new book out you might like, by Kamathan Packer, called Feminism in the Wild.
Yes.
Okay, do you know it? Just came out. I'm very excited about it. It's a really great book. I've been kind of speed reading it the past few days.
She has a very nice thank you to me in the acknowledgements at the beginning. So yes, I'm very
awesome.
familiar.
Great.
I
recommend it for everybody on this chat. I think you'd really like it.
I helped to spread the word that the book is out. So
excellent.
Yeah.
Good.
Thanks.
Yeah.
Cool. Thank you, Suleima. I'm gonna go perfect, and after I guess it's Karsten and then Erin.
Hello everyone, my name is Perfect. The research manager at the Succulent Karoo Research Station, and I'm currently doing my honors with University of South Africa. So it's a degree between Bachelor of Science and Master's degree. And I'm also doing a study on the activity budget of the Diurnal Bush Karoo Red by camera traps. And thanks for the talk, Vince. And my question is, I wanted to ask, how do environmental factors, for example, predation, interact with age structure to shape social network dynamics?
Yeah, great question. I don't know the answer to that. I think one of the problems is I can sometimes find the bodies of the beetles that have been eaten. And so I can sometimes guess what predation is like on a population, but I think in order to really get at that question, I'd have to do small mammal trapping, and that would require me to get all of the approvals to do all of that, as well as do something I'm not very good at. And so there is some small mammal data that's being collected about 2 miles down the road. And so I think I'd like to look at that and say, at least in temporal variations in small mammal years, do we see differences in age structure? But I don't know. I suspect that in high predation years, the following year you're going to get a younger age structure than in years when you've got low predation, but I don't have those data yet. Yeah, so I don't know what's driving this kind of big differences in age structure that we're seeing among the wild populations. Might be the age of the population. It might be predation. It could even be something like we have dry years. We had 1 year where it was extremely cold winter, and it knocked the population back to really low levels. And so there could be something even like temperature and humidity fluctuations could be structuring some of these age things. I don't know. But what I do think is that these ecological factors that might be driving it, this could be a really interesting and fun connection between ecological structures, ecological factors, demographics, and social behavior. Because if ecological factors structure age structures of populations change social behaviors, it could be kind of a fun area to explore how ecology drives evolution of social behaviors and weaponry. But I'm just not there yet.
Okay, thank you very much.
Yeah, thanks.
Yes, my name is Carsten Schradin. I'm a director of research at the CNRS in Strasbourg, France, and director of the Succulent Karoo Research Station, studying mammalian social evolution, especially in small mammals. It was really a great talk, very fascinating what you can do with these beetles. I have 2 questions. I will focus on the most obvious one. I'm surprised wasn't asked, which probably means I didn't listen carefully during your talk, which focus on the effects of age on different parameters of social networks and fitness. But when you look at your graphs, it's really the 1-year-olds that stand out to all the others. Sometimes it's also 2 and the other ones, but it's mainly the 1-year-old ones. And I assume from your methods, they're not really 1-year-old, but this is the year they have been catched for the first time. So they're actually younger than 1 year. And I was wondering how you can explain this and whether it might be that because these beetles just came out of their pupae, there's an ontogenetic factor like that after they emerge as adults, they first have to spend time and energy into spermatogenesis and oogenesis, and then an ontogenetic factor like this could explain both differences in reproductive success and social behavior in these young beetles compared to older ones.
Yep. You've hit on the biggest weakness of the study, right? So these are first year beetles. So they're the first year they're captured. They've eclosed from the fungus, and they've spent several years as a larva, but you're right, this is their first year after they pupate. And so that's something I really need to nail down. The thing is that in the lab, when beetles eclose, once they've hardened, so when they first eclose, they're like this really nice red amber color, and then their carapace oxidizes and they become harder. Once that hardens, they're fully sexually mature. I can mate them in the lab and they produce viable offspring that they lay eggs. I can take unmated females, put them with new males, vice versa, and I get eggs. So they're at least sexually mature. But it could be, rather than something like a middle-aged effect or reproductive improvement, it could be a difference, like you're saying, something physiological about they're not fully mature or something that's kind of preventing them from reaching that higher level of reproductive output. I go back and forth between being kept up at night about it and then saying, I'm not sure it really matters because there are differences in age regardless of what the reason is. They're sexually mature. So it's not that I'm looking at juveniles and adults and being like, oh, there's age differences. But it could be something similar to that. So yep, that's something I worry about. And so I think what I need to do is yeah, I'm not sure the way to answer that aside from doing what we did, which was try to make sure they're all sexually mature and that there's not some tail end that's not, which they are. But I'd be happy to hear any other suggestions you have on trying to rule out that concern and whether you think it matters or not. Yeah.
I mean, it does maybe not matter. I mean, if you find a difference in age at some stage, anyway, have to find the physiology explaining it. So there must be something. Different age must represent something in physiology. Yeah.
Yeah. Yeah. Interesting. Thanks.
Okay. Thank you.
Yeah. Thank you.
Thank you, perfect. And Karsten, Erin.
Hi, Vince. Hi.
Erin.
Thanks for a really great talk. So I'm Erin Syracuse. I'm a postdoc at the University of Exeter, and I've been working on these questions about social aging and sort of what are the causes and consequences of it. So it's really fun to hear you talk about your work because I've read a lot of your work and I've read a lot of Phoebe's work. So yeah, really fun for me to see it presented. I'm just curious about what you know or what you can speculate about the sort of mechanisms. So, and some of this might be me not necessarily remembering exactly which sexes you were presenting in the networks. But my recollection is that you were talking about males who have stronger connections to other males have increased fitness. Females who have stronger connections to males have increased fitness. And I'm just curious, what do you think is going on there? Like mechanistically, how are those fitness benefits coming about? Why are those stronger connections? Especially that I'm thinking the stronger connections between males, where you would expect that to be sort of maybe detrimental given strong agonistic interactions. But also, yeah, I may not have remembered the sort of which sexes you're talking about in the networks exactly. And did you talk about female networks at all? I don't quite remember that.
Yeah, no, I did not. Those are great questions. And I think they harken back to me, the things that do keep me up at night, which David asked right away, kind of got right to the heart of the matter is what are these things represent? What are the behaviors that are leading to it? And not, so that's one direction, like what are the behaviors you're actually measuring, but then the consequences also, like you're suggesting, are also kind of hard to sink my teeth into. So in previous work that I didn't talk about, when we changed the ecology, we changed the distribution of the resources, females had lower fitness in tightly connected networks. So it turns out that if you make the networks more tightly connected, females on average have lower fitness. So it really is kind of group selection there in that demographic factor really changes their fitness. And so we've always been kind of assuming that it has to do with male harassment of females because of this courtship and dancing. Females are often walking around with these males dancing on their back. And then in some of the populations early on that we measured in males, interacting with males had negative consequences. And that's what we saw in the ecology experiment too. So it was just like a nice, clear explanation, right? It's like male aggression, male aggression, female courtship, or female harassment. But in this study, in Phoebe's study with the age, right, we saw the opposite. Being more connected was really good. And so I don't know. Wild speculation might be about information flow. So knowing where the females are, where the good oviposition spots are, where those social hubs are, it strikes me as as very hand wavy. And so I'm not sure if that's what's going on. But that's kind of what I was thinking might be happening. Also, with these 1-year-olds, these females were pulling out of these cages. It might be different because they may be sperm limited. And so maybe they're laying more eggs if they're mating more and getting more sperm, especially the younger females. And so it was the younger populations, if I'm remembering right, that had that increase. Oh no, it was the older population. So that's not a good answer. Yeah. So the answer is I don't know. Yeah. Why interacting with more males would help you? Yep.
Yeah.
No, great question.
Yeah.
Don't know a
Okay.
good
No, I
answer
mean
to
that's
it.
totally fair. I
I
mean, this
beat
is
my head
something
on
that
this. I
we're.
gave Phoebe such I gave Phoebe such grief for this. I was like, come on, in your discussion, you really need to push yourself. And finally, she's like, you need to help me. And I was like, okay, this is a hard problem. I don't really know. You're right. This is tough. What is the explanation? And she's like, I'd rather not just wave hands. I was like, okay, you're right, Phoebe.
Yeah, I mean, I think it's a problem that we're grappling with across systems still. Is like, yeah,
Yeah.
great. We know that there are these connections between sociality and fitness, but why those exist still, we're is a big question mark in a lot of cases.
Yeah, and I beat my head against it too because I'm doing these experiments. I was like, haha, I'll know mechanism. And like, nope, I'm still just showing patterns. I need to do more. So
Yeah.
so we are doing courtship trials, one-on-one courtship trials between males and females and looking at preferences for age and courtship investment. And then we're also doing combat trials. And I was really hoping females would have combat, but they don't. And so I don't really know. Oh, your other question was about female-female networks. When we
Yeah.
put those together, they look non-random, or they look random, unlike joint sex networks and male-male networks. And then in that experiment with the ecological experiment, we constructed, after we ran the main model, we constructed male-male networks and female-female networks and looked at fitness consequences. And so what we saw is that male-male networks have fitness consequences, your location in the male-male networks. Your location in the joint sex network also has consequences for males.
Okay.
For females, your location in the joint sex network has fitness consequences.
Yep.
But when you make female-female networks, there's no fitness consequences. So that's a post hoc removing, constructing different networks from the same question, but it's indicating that there's something about interactions with males, regardless of your sex, that are driving these fitness consequences.
Yep. Well, I think that's
Small.
interesting in and of itself. But yeah, thank you.
Thanks. Yeah, thank you.
Thank you, Irene. Okay, so I guess it's gonna be me. So I'm Adriana Maldonado. I'm a professor at the Universidad del Rosario, and I study how social relationships form and how they are maintained or respond to disruptions of the environment. And I was indeed surprised that Erin didn't ask about this, but since you studied the aging part, but in your presentation, Vincent, you mentioned or you showed that individuals are less social as they age. And we know that, I mean, it's been shown in primates that it happens and some other species as well. And it seems to be something related to, protection from diseases. Could be, Erin can talk a bit about it more. But what do you think, or what are your thoughts on why does this happen in this population of beetles? Why do they become less social?
Man, you guys are asking all of the tough questions. Yeah. The answer is, like everything else I keep saying, is I don't know the answer to that. This is a remarkably consistent finding across species. So we were really excited to see it in beetles because a lot of the hypotheses, like in marmots and primates, have been about that individuals, especially older individuals, are decreasing kind of potentially frivolous connections or connections that don't matter very much and increasing investment in kind of more meaningful connections, right? And so in beetles, I don't think that's what's going on. I have no evidence for it. So I was wondering if it's, considering it's the same pattern, I was wondering if it's something more about decrease in movement. And so they're just not traveling from bracket to bracket as much. So maybe there's physiological wear and tear. But considering there's other improvements in performance, that doesn't seem very plausible. So one way we could look at that is, again, like someone else suggesting, tracking movement from bracket to bracket to bracket, because we record location. And in the beetle areas, I've got X and Y coordinates on this artificial log. I know exactly where they are. It does look like older individuals do have smaller home ranges, but that doesn't mean they're not like moving smaller distances. So that's something I think would be a good way to get at whether it is something about like travel or something. That they do have fewer connections, and And so I need to think a little bit about whether they're strengthening the connections they do have, which is a hypothesis we've seen in marmots and primates as well. So that's something I should go back. I'm taking notes about these sorts of things, go back and look at. And it's clear to me from these questions that digging into the behavioral mechanisms is something that would make the story much more compelling. And so looking at movements and home ranges, I tend to avoid those questions because the papers end up being very species specific, but would really help with the story. So I appreciate all these questions. Yeah.
Yeah, I mean, that's one mechanism in my work in different systems, right? Probably not necessarily in primates or marmots, like it wouldn't apply for it, but other type of beetles or insects in general are like, and since you already mentioned it, I'm going to just go for a follow-up. So we see this increase in reproductive performance, which also goes down as you age. So you have middle age, it has a good fitness. But if we assume that the sociality, it's somehow driven by a reduced space use, let's call it like that, it might be related to something physiological. Like maybe they don't have enough nutrients as they age. Or I don't know, do you have any thoughts on that? Have you given any?
No, I have not thought about like energy inputs or nutrients. They're all in the same environment, but you're right, if it's physiological, so they're just not able to, say, digest ligand as well as when they get older. I would be surprised by that, given that males who are older, you mean that decrease as from 2 to 3-year-olds? Yeah, that's interesting. No, I haven't thought about that very much because this experiment was the first to show that decline. We saw a little bit of it in the wild, but I haven't thought very much about why you're getting that, what might be senescence in that 3-year-old cohort. And our 4-year-old cohort in the wild was really, really small, so we didn't get a good look at that. No, I'll have to think about that if there is senescence, some sort of physiological senescence in that 3-year-old cohort. Last year, our 2-year-olds we didn't have very many produced, probably because of the cold snap. And so we had a lot of 3-year-olds. So I'll have to go back and look at that data that we did for like fighting and movement. We did some open field tests and see if there is just movement in that way. Might be interesting. Yeah.
Cool.
Yeah, I like that.
Have you noticed if they are less, they have decreased competitive ability at the age, or?
So it depends. In general, it looks like they have increased competitive ability. Older males, from a cross-sectional analysis in the wild, older males were more aggressive. They initiated more fights. Recent data from our captive beetles shows that 3-year-olds are much more likely to win combat than 1-year-olds. But because our captive beetle populations fluctuate in our productivity from year to year, like what would be nice is to have 1-year-olds, 2-year-olds, 3-year-olds, and 4-year-olds in captivity and do all of that. But when you've got a polar vortex that comes down, you lose your 3-year-old cohort, and now next year your 4-year-old cohort's small. And so we every year we've got 2 ages usually that we can compete. And so, it's kind of piecing together these pieces. But it looks like, in general, older individuals are more aggressive and win more fights, at least for males. Females don't seem to show any conspecific aggression that we can measure anyway.
Cool. Thank you. We have a question from Gary.
Hi. Thank you. Thank you for the talk. It was really nice. And I'm Gary Moura Delgado, and I'm currently working at CBIO. It's a research center in Portugal, and I'm investigating the fitness trade-off of social network metrics and sociability in sociable weavers. That it's a cooperative birds endemic of Southern Africa. And I had more like a comment more than a question, and it was that why I was waiting till the end. And it was in relation to David's question and comment. And it's also because I found that there is some relation between the sociability of the sociable weavers and their survival. So social metrics and survival. So there is something that it's affecting fitness. And I really don't think that we can only see social metrics as a component of those basic trait or morphological traits. And also the suggestion that those are the only ones that can be heritable or have heritability. I think that if we follow that reasoning of these social traits having the possibility of breaking them down, I think it could work for any morphological trait in reality. For example, if we think about body size, we can also see that or think as body size as a component of, for example, hormone, growth hormone, or mitochondrial activity, or a compound of physiological traits. And that body size also depends on other environmental factor. And I think that it's totally fair that think that even body size is a component of physiological traits. At the end, what matters is that body size might be subject of selection. So I think that is the same for social traits, that maybe they are a component of any other basic traits or morphological traits or whatever you want to call them. But at the end, if there is an effect of social fitness, of social metrics on fitness, at the end, what can be a subject of selection are those social metrics traits. And I think that they can be evolved, and it's totally fair also to focus on the heritability and the evolution of those social traits without focusing on those basic traits that can be compounding them. I don't know. I don't know if you have anything to say about it, David. It's just question was a comment. More a comment and a thought than a question, but I think it's fair to also to focus on the heritability of these social traits, even though we think they are a component of other, because at the end they can be subject of selection as body size can be equally.
Yeah, I mean, I think this is not necessarily a divide among social network folks. Personally, I swing back and forth. I think these are emergent properties that are the product of social interactions among individuals. If they're under selection and we can show there's some level of heritability, probably will involve indirect genetic effects. And so like it can evolve. Do I really care if it's emergent or not? And I'm just like in that camp. And then there are other times where I'm like, nah, I suspect these are just the product of, linear combinations of traits, and we can distill it into those things like body size, right? You could start to explain some of the variance in body size by looking at growth hormone, right? And so you could study growth hormone and be interested in that. And so I swing wildly back and forth into these different perspectives. And I think it's all interesting, and I think it all leads you to good questions, right? So if you're really interested in breaking down the behavior into its components, I think that's going to lead you in interesting worlds. I think if you're just like, I don't care, it's an emergent property, it can evolve, we know indirect genetic effects exist, and I think it can evolve, it's at a different scale. I think that's going to lead you into different camps. And I'm not sure that you're going to be right or wrong in either way, right? Because I suspect there's elements of both of those things are true.
Yes,
somebody, thinks across
that
scales.
both questions are totally fair, that we can focus on breaking them down and try to explain why it's it and which is the things that are making that up. But I think that focusing also directly on the social traits is also totally fair, yeah.
Yeah. Yep. And I think, yeah. And I think that whether it's a reductionist approach or an emergent approach, I think you're going to find out interesting things. And I really do just chew on it both ways. I do think what I found is when I give these talks, I've been talking about this for a long time, and my first knee-jerk reaction was like, it's emergent, it's cool, IgEs are going to solve our answer, it's okay. People really find it not satisfying, right? And they want to know what these things represent because they want to understand it. And so I think that has pushed me to try to be more reductionist in my thinking, because it may just be more compelling to people if you can give them something that they don't need to like hold separately in their mind. And so I think that questions often push me to be more reductionist. And so it's nice to hear somebody be like, don't worry about it. Keep going. Yeah, so thanks for that.
Thank you, Gary. And our last question of the session, it's Sulaiman.
Yeah, I just had a couple more thoughts that I think they're not really questions. They're just thoughts, excuse me, of things that might be worth thinking about. And
Thanks.
there's two things. The first one is that you have assumed that the unmarked beetles that appear after you've marked the whole population are beetles that just eclosed, and so therefore they're young first-year beetles. But the other possibility, of course, is that they're beetles that are dispersers immigrating into your study population. And I don't know if there's any, I mean, it's the bane of the life of anybody who works on dispersal in the wild, that you really often can't tell between mortality or, new and dispersal or new animals that appear, and whether you just didn't manage to trap them before, and then you suddenly trap them and they've been there all the time, or whether they just dispersed in. But it might be worthwhile to think about that, and maybe even if it's only in modeling, compare if there's any difference between the assumption that you're currently making versus if that assumption is not correct, and instead there's adult beetles that are dispersing from elsewhere, even perhaps from beyond, since they can fly, beyond your study colonies. And then, so that was one thought. The other thought, I don't mean to beat a dead horse, but I couldn't help thinking about the females interacting with more males, having higher reproductive success, and coming back to my question about the Bateman gradients for those females, and whether in fact there may be something going on. I mean, if they're and again, this is where actually having the behavior would be really good, the nitty-gritty of the behaviors. When females are interacting with males, what do those interactions consist of? Like I said, like, hello, how are you? Goodbye. Or are there interactions that in fact involve mating, in which case it at least raises questions as to whether female reproductive success could be affected by number of mates or interactions with, assuming interactions are matings with other males, then there could be something there that we're missing. And I'm going to try to send you several papers that I think you'll find interesting. I'll email them to you.
Oh, thank you very much.
Throw that out to see if, I just thought it might be worth thinking about.
Thank you so much. Yeah, I really think it is. I'll hit a couple of those points real quickly. One is that you're absolutely right. Those could be dispersers instead of young. We try to mark everybody in the forest within double the median dispersal distance, but absolutely we could just be measuring effects of like really long distance dispersers, right? The nice thing is that in the lab populations, in the beetle areas, those are beetles I know the age of because I know when they were laid, right? And so I know the age, and we get the same results in the beetle area, so that makes me feel a little bit better about it. But you're right, in the wild, that's certainly the case. The Bateman gradient for the females is really an interesting idea, and I could do that too because I know who they mated with and how many eggs they laid. So that would be a really easy regression to run and see what we're getting. Yeah, and I'm sure that quality of sperm and quality of the eggs changes as individuals age, and I haven't been able to look at that yet, but is on my list of things to do when I've got some incubator space. Yeah, so all of that all of that is really interesting. One of the things we're going to do to get a closer look at behaviors is run a bunch of behavioral trials in all 400 individuals. So like mating trials, aggression trials, and open field tests, and then phenotype everybody and then put them into the beetle areas. And that should help a little bit. I think the other way to do that is to get students to do focal follows, which is remarkably boring because these beetles move really slowly, like really, really slowly. Lily can attest that if you just sit there and watch these beetles, it's not very fun. And so I've got to, figure out the best way to do these focal follows that'll get us a sense. Right now, it's all proximity. So it could just be, hey, how are you? It could just be beetles being like, oh, male, and turning and walking sideways, or it could just be nothing, random, associations. So thanks for all of that, and I look forward to the paper. Thanks.
Yeah.
Well, thank you, Vince, and thank you everyone really for such a great discussion, and Vince for such a great talk. Really interesting, and we really hope to hear back from you with all these questions that we have asked you.
I've written them down.
Yeah.
Great. Thank you so much, everyone. Really enjoyable time. Discussion was fun.
Okay, well, thank you, everyone, and we'll see you next week.
Yeah, and I hope you come back, Vince.